Now we proceed to the pre-treatment of slide before probe application. This step is required if the slide has cellular debris or red cells or proteinaceous deposits. This step can be avoided if the slide is clean and is prepared from harvest. Keep the slide in 2x SSC buffer for 15 minutes. The slide is gently agitated 2 to 3 times in between. After 15 minutes, we take out the slide from 2x SSC buffer and proceed for pepsin digestion. For pepsin digestion, transfer the slide into pepsin for 6 minutes. Again, the slide is gently agitated 2 to 3 times in between. This incubation time is to be adjusted based on the amount of cellular debris in the slide. After 6 minutes, to stop the pepsin digestion, transfer the slide in 2x SSC buffer for 5 minutes. The slide is gently agitated 2 to 3 times in between. After this, we proceed for gradual dehydration of cells. For gradual dehydration, transfer the slide in 70% alcohol for 2 minutes with gentle agitation in between. Then transfer the slide in 85% alcohol for 2 minutes with gentle agitation in between. Finally, transfer the slide in absolute alcohol for 2 minutes duration. The slide is gently agitated 2 to 3 times in between. After 2 minutes, take out the slide from absolute alcohol and air dry the slide properly. After the slide is air dried properly, the slide is now ready for probe application. Now we move to application of probe. Probe application should be done in dark. Take out the probes from the deep freezer and briefly spin them in mini centrifuge before application. Probes are either in diluted form, for example, Zytovision metasystem probes, or needs to be diluted before application, for example, Weiss's probes. Dilution of probe is done by mixing 1 microliter of probe with 2 microliter of distilled water and 7 microliter of hybridization buffer. The diluted probe can be kept at -20 degrees centigrade for up to 3 to 4 months. In this demonstration, we are using a pre-diluted probe. Before application of the probe, we should be ready with clean cover slips of appropriately sizes. Inappropriately larger cover slips will require more probes, which otherwise leads to bubble formation or drying. Cover slips are cleaned using absolute alcohol and properly air dried before application. Appropriate volume of respective probes are applied to the marked areas in the slide. For example, in this particular marked area, we require 2 microliters of the probe. Avoid bubble formation and spillage of probe. Now, gently cover the area with cover slip. Air bubbles, if present, can be removed by gently pressing with the blunt end of the forceps or by a micro tip. Completely seal the cover slips using rubber sealer after ensuring that there are no air bubbles inside. The slide is now ready for hybridization. We start the hybridization process. Switch on the hybridizer. Ensure adequate water in the water chambers. Select the appropriate hybridization cycle. We do denaturation at 78 degrees centigrade for 2 minutes, followed by hybridization at 37 degrees centigrade overnight for 14 to 16 hours. This needs to be modified according to the manufacturer instructions. Keep the slide in the hybridization chamber. Close the lid and start the hybridizer.